Okay, so last time we finished up talking about myelination and unmyelinated axons and how they differ. Now we're going to get into axon regeneration. Okay, so after some traumatic injuries, whether that is maybe a physical thing or typically when we think traumatic injuries, that's going to be a physical thing, but it could also be some type of viral or bacterial infection. After some kind of injury to the nerve axons or to the nerves, the peripheral nervous system axons can regenerate themselves. So it's only possible if two things happen. If the neuron cell body is still intact, so the soma still has to be intact, and enough of the neurolemma has to remain to be able to fully regenerate. So if you remember, the neurolemma is what is left over after the axon has been myelinated, after the neurolemmacyte wraps around and around the axon, the cytoplasm and the nucleus get kind of pushed to the outside, and that becomes the neurolemma. So as long as there's enough of that left, then regeneration can be possible. Regeneration will be even faster. It's more likely to happen if the amount of damage is less extensive, which makes sense. The less damage, the easier things are to repair. And the distance between the site of damage and the structure it innervates is shorter. So as long as the neurons aren't going to be these long axons, as long as it's just very kind of short distances that they're having to repair, then regeneration is much more likely to happen. And that's because, as we'll see within the steps, that axon actually has to repair itself. So what are the steps to it? So the first step is that the axon is damaged by trauma, severed by some type of trauma. Proximal to the cut, the axon seals off and swells. So when it says proximal to the cut, it means that the part closer to the cell body. So the part of the axon that is closer to the cell body, that will remain, but the part that's distal from that, the axon and the sheath degenerate. They'll actually start to break down, called Wallerian degeneration. But the neurolemma still survives. So remember, the myelination and the neurolemma, they wrap around the axon. So the axon and the sheath itself will degenerate, but the structure, kind of the scaffolding for it, is still there. What ends up happening is that the neurolemma and the endoneurium form a regeneration tube. If you remember, the endoneurium was that outer connective tissue layer of the axon. The axon regenerates, guided by nerve growth factors released by neurolemmacytes. So neurolemmacytes release types of local hormones called nerve growth factors that will encourage that growth. The axon re-innervates the original effector or sensory receptor. So after full regeneration, remember, regeneration is the restoration of functional ability. So after full regeneration, the axon is able to re-innervate the original effector. Here is just a diagram of that, which I think is a little bit easier to follow. So you can see right here you have some type of traumatic injury that's severing the neuron. The proximal portion is severed and swells. So again, proximal to the cell body, it's going to start to kind of get a little bit more swollen. And then the portion distal to that will start to degenerate and break down. Eventually you end up with just this regeneration tube. That remyelination begins to occur as its growth factors start to take over, and then eventually innervation to the effector is fully restored, hopefully. In the central nervous system, axon regeneration is extremely limited. The oligodendrocytes actually exhibit or release growth inhibiting molecules rather than growth stimulating molecules. And the number of axons in the CNS, there's just a very much more limited space. So there's just not as much room for these axons to be able to develop and regenerate. And also, regrowth is obstructed by scars from astrocytes and connective tissue. So basically, once there is a space, other things will start to fill in there, and they'll have to get bumped out of the way, and usually that's not going to happen. So regeneration in the CNS, you don't see it as much. Also, that is partly why the central nervous system is so much more protected. It's housed within the skull and the spinal column versus the peripheral nervous system, which is going to be more outside of that. The peripheral nervous system is easier to repair, whereas the CNS has more protection, but it does not repair as easily. Okay, so now getting into more of the axons, the types of pumps and channels that we see there. So pumps, if you remember, are membrane proteins that maintain a concentration gradient by moving substances against their concentration. Maintain a concentration gradient by moving substances against their concentration gradient. That requires cellular energy. If you're going to move something against its gradient, it requires ATP. And neurons have sodium, potassium pumps, and calcium pumps in their membrane. So those are going to be the two primary ones that we'll see. There are channels. Channels are protein pores. I'm going to shrink myself a little more. Maybe not that much more. There we go. Channels are protein pores in the membrane that allow ions to move down their concentration gradients. That could either be into the cell or out of the cell. We'll see that with sodium and potassium, they're moving in different directions through these channels. When they are open, they allow a specific type of ion to diffuse. So you're going to have a sodium channel, you'll have a potassium channel, you will have a chloride channel. They won't be moving through these channels just at will. Leak channels are passive channels. They're all going to always be open for continuous diffusion. Remember, diffusion is something moving down its concentration gradient, and it's a passive, easy process. Chemically gated channels, normally closed but open when the neurotransmitter binds. So a chemical gated and a neurotransmitter, they have to go together to be able to function and open. And then voltage gated channels, they will open when membrane charge changes. Otherwise, they are going to be closed. Okay, so talking specifically about the voltage-gated sodium channels, because those are pretty important channels and they have an interesting mechanism for operation. So voltage-gated sodium channels, they have two different gates. They have an activation gate and an inactivation gate. And essentially those two gates, one is going to be exposed to the cytosol, one will be exposed to the interstitial fluid, and as they open and close, they have to kind of work in concert to allow ion flow in and out of the cell. So the voltage-gated sodium channel exhibits three different states. It has a resting state, an activation state, and an inactivation state. And it cycles through these very, very quickly. So it's important to remember when we say resting state, we're talking about moment zero. We're talking about, just being able to observe it. It's hard to actually, in real life, look at a person and say, okay, now you're at rest and now you're working, because our neurons are constantly firing. We're constantly sending signals, regulating things. So the resting state is very much a clinical thing that we talk about. So in the resting state, your activation gate will be closed, but your inactivation gate will be open. And that still prevents the entry of sodium. So even though the inactivation gate is open, because the activation gate is still closed, you won't have any sodium that is able to come in. And the activation state, the activation gate becomes open because of a voltage change to the membrane potential, and the inactivation gate is also open. So that allows sodium to move through the channel. So in your activation state, your inactivation gate stays open, your activation gate opens up as well, and sodium can flow through. Then in the inactivation state, the activation gate stays open, but the inactivation gate closes. I know this starts to get a little bit confusing. So just we'll see a picture of it in a moment, and just kind of trying to remember that, okay, those two gates, they're going to be opening and closing relative to the voltage stimulation that they receive. So during this activation state, both gates are open, but this is just for a brief period of time. This is not for a long time. This is just for a brief period of time. And then in the activation state, the activation gate stays open, the inactivation gate closes, and that, again, prevents the entry of sodium, because if the inactivation gate is closed, nothing is going to happen. This, again, state only lasts for a short period of time. It quickly resets to its resting state. Here is a picture, which, again, is always a little bit easier. So you have the three states, resting, activation, and inactivation. You have interstitial fluid, your plasma membrane, and your cytosol, so outside of the cell and inside of the cell. And you can see here the two gates. So you have your inactivation gate and your activation gate. So in the resting state, this inactivation gate is open and the activation gate is closed. You're at your resting membrane potential, no voltage to stimulate it. In the activation state, you've received that voltage, so you can see that the charge on the membrane has changed, has switched, so now it is activated. In the activated state, both gates are open. This is going to stay like that for however much time until the cell starts to hyperpolarize, until the cell reaches threshold, and then the inactivation gate starts to close. You switch into this inactivation state, and the activation gate remains open. This only lasts for a brief period of time, and then it switches back to this resting state. So let's watch this video just to give us an idea of that. Actually, I already have it loaded up.
When the cell membrane is at its resting membrane potential, the activation gates of the voltage-gated sodium ion channels are closed and the inactivation gates are open. Voltage-gated potassium ion channels are closed. Depolarization is initiated by a stimulus which makes the membrane potential more positive, causing the voltage-gated sodium ion channels to start to open. As threshold is reached, many sodium channels open. Sodium ions diffuse across the membrane, causing depolarization. Voltage-gated potassium ion channels also begin to open, but more slowly. Therefore, depolarization occurs because more sodium ions diffuse into the cell than potassium ions diffuse out of it. As the membrane potential approaches maximum depolarization, the inactivation gates of the voltage-gated sodium ion channels begin to close, and the diffusion of sodium ions decreases. The potassium ion channels remain open, and potassium ions continue to diffuse out of the cell. The increased potassium ion permeability lasts slightly longer than the time required to bring the membrane potential back to its resting level. The extra efflux of potassium ions causes the membrane potential to become slightly more negative than the resting value. After the voltage-gated potassium ion channels close, the active transport of sodium and potassium ions reestablishes the resting membrane potential.
Okay. So hopefully that kind of gave you a little example or a better idea of it. So remember, these are
communicate with each other through their dendrites, membrane.
There we go. Okay, so remember, these are voltage-gated channels. So as the voltage shifts, as the movement of ions changes the voltage of the plasma membrane, that's going to cause the gates to act differently. Okay, so watch that video again if you need to, read this more through your book, and definitely ask me questions if this doesn't really make sense. Voltage stuff, electrical currents, we're going to get into that with this chapter. It's tricky. It's kind of weird if you're not okay and comfortable with the idea that the movement of charged ions also creates electricity and how that affects gates. It's kind of strange. So let's try to get through this chapter, and hopefully learning all of it will help. Okay, couple more types of gates and channels. Modality-gated channels, they're normally closed, but they open in response to a stimulus other than chemical or voltage. Typically that is going to be some kind of change in environment, like pressure. So y'all can't see me down there. So like, pushing down on something, that's going to activate these modality-gated channels. As you push down hard, as they have more stimulus, they will fire even more. They will open even more. Okay, so how are these pumps and channels distributed throughout the plasma membrane of a neuron? Because we'll see that there are different parts of the neuron, and they all are going to function slightly differently. And form follows function, so they have different channels and pumps. So the entire plasma membrane of the neuron is going to have leak channels, those potassium and sodium leak channels. It will also have sodium and potassium pumps, and those are really going to be, we'll say this over and over again, the sodium and potassium pumps are there to help maintain that concentration gradient. We'll see they only contribute a small amount to the resting membrane potential, but it's that concentration gradient that they really help with. Okay, so we can break the neuron, and when we're talking about the distribution of pumps and channels, it's important to break the neuron down into its different sections, or the parts of a neuron into its different sections. So the receptive segment are the dendrites and the cell bodies. That is full of chemically gated channels, like chloride, potassium, sodium gated channels. And again, that makes sense because that's going to be the receptive segment. It's the dendrites, it's the cell body that is receiving the signal. Remember, a neuron releases neurotransmitters, which is a chemical. So having chemically gated channels at the receptive segment allows for them to accept the signal. The next part is the initial segment, so that's that axon hillock. So between the cell body and the axon, you have that little portion called the axon hillock. That has voltage-gated sodium and potassium channels. The conductive segment also has voltage-gated sodium and potassium channels. The conductive segment is exactly what it sounds like. It is the length of the axon. It's conducting that signal down the axon. And then the transmissive segment is the synaptic knobs, the synaptic terminals, synaptic bulbs, whatever term you want to use for them. That's the very end portion that is going to be sending the signal or passing the signal to another neuron's receptive segment. So the transmissive segment is going to have voltage-gated calcium channels and calcium pumps. If you remember, when the action potential arrives to the synaptic knobs, that is going to open up those calcium channels so that calcium can diffuse into the neuron and allow the neurotransmitter to be released. So it's important for calcium channels and calcium pumps to be in the transmissive segment, so that way it can, the calcium pumps are going to make sure that the concentration gradient of calcium is lower on the inside of the neuron than it is on the outside of the neuron. And the calcium channels will allow calcium to flow in. Okay, so calcium channels allow stuff to come in. Calcium pumps are going to push the calcium out to maintain concentration gradient. Calcium is necessary for neurotransmitter release. Here is half of that picture. So you have plasma membrane, the entire neuron. You're going to have these sodium-potassium pumps, sodium leak channels, potassium leak channels. In the receptive segment, you're going to have your chemically gated channels. Okay, so in receptive parts, that's where the neurotransmitter is released. You have to have chemical gated channels for those. In the initial segment and the conductive segments, those are going to be voltage gated channels. Because remember, the neural signals, we're taking an electrical signal, we're transmitting it to a chemical one, and then we're turning it back into an electrical signal, and then it'll be turned back into a chemical signal. So these chemical gated, voltage gated channels, if you kind of think about it like that, it makes sense where they're going to appear. So the initial and conductive segments are all voltage gated. Real quick, just while we're here, remember the sodium-potassium pump, that maintains this concentration gradient. And these sodium channels, these leak channels, they help to maintain resting membrane potential. So having a little bit of potassium leaking in, a little bit of sodium leaking in, helps to maintain the proper membrane potential. Then at the transmissive segment, the very last portion, you have the calcium channels and the calcium pumps. Okay, remember like we said for neurotransmitters. Okay, so neurons and Ohm's Law, getting into a little bit of electrical physics here. So electrical energy, this goes back to our first couple of chapters. Electrical energy is the movement of charged particles. Neuron activity is dependent upon electrical current. So remember, charged particles, those are going to be, typically when we think about electricity, we're thinking like, especially like in a battery, those charged particles are electrons. And the current is how electrons flow through the battery, and that creates a current. When you put a battery into something, that battery is inserted into the current, so that way it can power. I have a light kind of behind me right now. That light is plugged into a socket. That socket is allowing current to flow through, electrons to flow through and power that light. So in neurons, the neuron activity is dependent upon electrical current. So our thoughts, our actions, all of our regulatory activity is based upon this electrical current, this idea that we can conduct electricity through our neurons to create responses. So there are three components to electricity that we have to discuss, we have to understand. Voltage is your potential energy. The voltage is the amount of difference in the electrical charge between two places. So if you think, again, we're talking about neurons, the plasma membrane is going to divide the two places. You have the inside of a cell and the outside of a cell. So the voltage is the difference between the inside and the outside. The current is the movement of those charged particles across the barrier, separating them. So the movement of charged particles across the plasma membrane will create some type of current. That current can be harnessed to do work. So remember, we said concentration gradients are potential energy, whereas actual movement of ions is kinetic energy. Okay. And then resistance. Resistance is the opposition to movement or opposition to work of charged particles. An increase in resistance lowers the current. So plasma membranes are fairly resistant to movement of ions. Remember, we said they have several different features that make them resistant to ion movement, which is why channels and pumps are so important and so necessary for resting membrane potential and neuron activity. So you have voltage, which is the actual difference in electrical charge. You have the current, which is the movement of those particles back and forth. And you have resistance, which is your plasma membrane. And I think that, yeah, here it does. It applies it to neurons here. Okay, so all of this can be summarized in Ohm's law. The law is current equals voltage over resistance. C equals V over R. What that means is that current increases with larger voltage and smaller resistance. So think about this as a math equation. Current equals voltage over resistance. That means that current and voltage are directly related, while current and resistance are indirectly related. So the larger the current that you have, the larger the voltage will be. And the larger your resistance, the smaller your current. The more resistance you have to flow, the slower things will flow across, the lower your current is going to be. So when we look at applying that to neurons, with neurons, charged particles are the ions, the sodium, potassium, chloride, calcium, and the current is generated when those ions diffuse through channels, when they diffuse through all those channels that we just talked about. The voltage exists across the membrane due to unequal distribution of those ions. So because we have concentration gradients, we have those pumps, those sodium potassium pumps and the calcium pumps establishing concentration gradients, there is a voltage difference across the plasma membrane. The membrane offers resistance to ion flow, and this resistance changes due to the actions of gated channels. So as those voltage and chemical gated channels open and close, they're going to change the resistance of the plasma membrane. If all of the channels are closed, then your resistance is at its maximum. If all of the channels are open, then your resistance, as it says, will really decrease. Okay, so characteristics of resting neurons. Remember this idea of a resting neuron, it's a moment in time. We are looking at just a glimpse here. So in a resting neuron, your ions are unevenly distributed across the plasma membrane due to the actions of those pumps. You have higher concentrations of potassium in your cytosol versus the interstitial fluid, so more potassium on the inside of the cell than you have on the outside of the cell. And a higher concentration of sodium, calcium, and chloride on the outside than on the inside. Your gated channels are closed in the functional segments, and there's an electrical charge difference across the membrane. So the cytosol is relatively negative compared to the interstitial fluid, and that idea of relative is kind of important. So the resting membrane potential is typically about a negative 70 millivolts, and you can measure it with electrodes. I think we do one more. Yeah. So it says relatively negative compared to the IF. We'll look in just a couple of slides at what is making the interstitial fluid so much more positive, what is making the cytosol seem so much more negative. I want to make sure I don't want to say anything else to this slide. Okay. Here's just a picture of this. So you have the cell body, your axon, and then your terminal segment. You can see as you go start up here, you have your chemical gated channels. As you start to get into the axon, the conductive segment, you start to switch to your voltage-gated channels, and then down here you have these calcium channels. So all pumps and channels switching slightly differently depending on what segment you're in. Okay, resting membrane potential. So again, a neuron at rest exhibits resting membrane potential. At rest, the potassium diffusion is the most important factor in setting resting membrane potential. Potassium will diffuse out of the cell due to its concentration gradient, but the diffusion is limited because of the electrical gradient. That pull of the negative resting membrane potential, the pull of the negative charge on the inside of the cell body will resist potassium's diffusion out. So this was chapter 2 or chapter 3 stuff that we talked about, how there's that electrochemical gradient for potassium, where its chemical gradient wants to diffuse out of the cell, but its electrical gradient wants to maintain it inside of the cell. And eventually, at rest, potassium kind of finds an equilibrium there. Never full equilibrium. It's never that potassium actually stops moving. It will constantly move, but it will start to find an equilibrium in that movement. The notes say, and they make a note of, that if potassium were the only ion leaked, the resting membrane potential would be around negative 90 millivolts. But because of those sodium leak channels, that allows a slightly more positive charge to creep into the cell, which puts it at about negative 70 millivolts usually. Oh yeah, and there that is. So sodium diffuses into its concentration gradient and the electrical gradient, and that small leakage means that it's less negative. That's really important for helping the cell to reach threshold, because if it were actually at negative 90, if there were no sodium leak channels, the neurons would have to work even harder, cells would have to work even harder to reach the threshold of an action potential. You have your sodium-potassium pumps. Remember, it's 3 positive charges out and 2 in. So 3 potassium out, 2 sodium in. It only contributes a small amount to the membrane potential, so really it's that concentration gradient that it's helping us with. Okay, so here's actually measuring that resting membrane potential. This is a voltmeter. They have microelectrodes set up. One is going to be in the cytosol, one is going to be over here in the interstitial fluid, and you can measure the voltage difference between those 2 areas, and you see it's set at negative 70. You have the sodium and potassium leak channels operating opposite of each other. You have sodium potassium pumps working as well to try and maintain that concentration gradient, and some other factors that are going to be causing that negative charge on the inside of the cell. Inorganic phosphate, proteins, those all contribute to the negative charge within the relative negative charge in the cytosol versus the interstitial fluid. Did we? I thought there was a slide about that. Anyway, but just remember then. I thought there was a note on that slide. Maybe it's coming up in another one. But the factors that contribute to the negative cytosol charge again are the inorganic phosphate and the proteins, because you just don't have. You're not going to have. Inorganic phosphate is necessary for ATP, so you need that inside of your cells. And proteins, if they're in your interstitial fluid, it should only be for a temporary time. There should not be a large collection or a large concentration of proteins in your interstitial fluid. That is a sign of something going very wrong. I'm not a doctor, so that's about as diagnostic as I can get. But if you have protein on the outside of your cells, you are in deep trouble. Excessive protein. Okay, let's look at the overview of the events in each neuron segment. We're actually going to end this lecture with the initial segment, and then the next one we'll finish up the rest of these notes for chapter 12. Okay, so in the receptive segment, which if you remember is going to be the dendrites in the cell body, this is where we see graded potentials. So I've talked a lot, and we started in the muscle chapter talking about action potentials. This time we're talking about graded potentials. So graded potentials are small, short-lived changes in the resting membrane potential with a couple of key characteristics. So small and short-lived changes. They're meaning that it's not going to be a big change in the resting membrane potential. It's only going to be a small one. And it's not going to be lasting for even seconds. It will be milliseconds that it lasts. Excuse me. So they're established in the receptive segment by the opening of chemically gated channels. Remember, there are only those chemically gated channels in the receptive segment. The local current changes only happen for a brief period of time, 10 to 15 milliseconds. And they vary in the degree of change and the direction of the change. That doesn't mean that it's going to, it's going to go left or is it going to go right or is it going to go up, is it going to go down. It's talking about, is it going to be an inhibitory or an excitatory change? Is it going to depolarize or hyperpolarize the membrane? The fact that it can be large or small, that's what it means by graded. Okay, so when it talks about a graded potential, means that the bigger the stimulus, the bigger the change. Whereas the smaller the stimulus, the smaller the change. And again, that direction of change refers to depolarization or hyperpolarization, either making it less negative or making it more negative. So we can talk about, in the postsynaptic neuron, the postsynaptic potentials. There are two different postsynaptic potentials. Remember, a synapse is where two neurons meet. So the postsynaptic potential is talking about the neuron in the receptive neuron that is getting the signal. So you can have an excitatory postsynaptic potential, an EPSP, or an inhibitory postsynaptic potential, an IPSP. If you have a postsynaptic potential that is going to depolarize your neuron, it will be excitatory. It will be depolarizing it closer to that threshold value, allowing it to transmit the signal. If you have an inhibitory postsynaptic potential, it will make the neuron, the resting membrane potential, more negative. It will hyperpolarize it and make it harder for that signal to be transmitted. You have numerous postsynaptic potentials that can be generated because a postsynaptic neuron can bind many neurotransmitters simultaneously. So you can have, we'll talk about summation in just a slide or two, but you can have 2 EPSPs and 3 IPSPs, and if the 3 IPSPs are stronger, your signal will be inhibited and it will not be transmitted. But you could have a 5 EPSP signals all coming because neurons, remember, they have those dendrites that can receive multiple signals, and as they receive those signals, they can kind of integrate them and they see, okay, well, what is the overall response here? So excitatory postsynaptic potentials are depolarizations caused by sodium entry. You see that you have right here this yellow bar, we have our resting membrane potential, you have a stimulus, and then you just have this little blip. again, it's going to be a small change. It's going to be a brief change. These are graded potentials. So it's not going to be an action potential. It's not going on its own. One signal, one EPSP on its own won't reach threshold. It'll just be a little blip, and then it'll go right back to resting membrane potential. So you have the neurotransmitter released. It binds to the chemical gated channel. Sodium will flow in faster than potassium flows out, which causes a slight depolarization of the membrane in that segment. If you have an IPSP, you're going to have the same thing. Neurotransmitter will bind a chemically gated channel. What actually happens here, as opposed to the movement of sodium, we're looking at the movement of potassium and chloride. So either potassium going out of the cell or chloride coming into the cell. Chloride is a negative ion. So whether potassium is leaving, whether you have a positive charge going out or a negative charge coming in, the result is still the same of a hyperpolarization of the membrane. So you can see you have your stimulus. You have this little dip, that's your IPSP, and then it goes back to the RMP. I know this kind of looks a little confusing, but just, it's okay, don't worry. What this is kind of just showing is just the numbers are just numbering the different segments, the different potentials that are arriving. If it's a red, it's an IPSP, it's an inhibitory. If it's green, it's excitatory. And what it's trying to say here is that you have all of these signals that are being received by this receptive segment. They're all moving into here, and then their collective action will dictate if the signal is transmitted. That collective action is termed summation, and we see that in the initial segment. So the initial segment, remember, is the axon hillock, and this is where the summation of the EPSPs and IPSPs occur. You have voltage changes from the dendrites and soma are all added together, and the sum may or may not reach the threshold membrane potential for initiating an action potential. So we started with the graded potential, which is small, short-lived in the receptive segment, and now we're starting to talk about action potentials. We're starting talking about taking all of those graded potentials, summating them, adding up their relative actions, and then seeing, okay, is this enough that's going to get us to threshold? If so, okay, we can create an action potential. If not, then there is no action potential. So threshold is the minimum voltage change required. It's typically about negative 55 millivolts, but that will vary depending on the neuron. And generally, you have to have multiple EPSPs to reach threshold. If you just have one EPSP, it's typically not strong enough to fully depolarize a membrane. If threshold is reached at the axon hillock at that initial segment, then the voltage-gated channels open and an action potential is generated. So summation can occur across space and time. I've been watching a lot of Star Trek recently, and so I'm very much into our space and time ideas. So spatial summation means that multiple locations on a cell's receptive surface are receiving transmitter signals simultaneously. Temporal summation refers to a single presynaptic neuron repeatedly releasing neurotransmitter, the same neuron, just releasing neurotransmitter over and over and over again, producing multiple, it just has EPSPs here. It could be EPSPs or IPSPs. You can have temporal summation of EPSP or IPSP. And this happens within a very short period of time. So you can either have kind of a mass movement where you have a lot of different neurons all kind of coming in and all saying, just kind of flooding with just a bunch of signals from a lot of different areas that can cause a threshold to be reached. Or you can have one neuron, a single presynaptic neuron, that is just, calling, knocking on the door, ringing the doorbell until it opens, until it's like, okay, I finally annoyed you enough that you're going to send the signal. You're going to open up and let me do my activity. Okay, so here is a spatial summation. Again, don't worry, it's okay. These graphs I know look kind of weird. But what it's saying is that you have two simultaneous EPSPs summed to produce a greater EPSP. So you have P1 is stimulated. So here is P1, and it's green, so it's an EPSP. And you can see it has a little blip. It goes right back to RMP. P2 is stimulated. It has a little blip. It goes right back to RMP. But when you have P1 and P2, when they're summated, when they're combined together, you get this even larger response. Okay? Same thing with the IPSPs. You can have P6 right here. You can have P7 right over here. And then individually, they have a response, but together, when they're summated, they're even larger. Now, if we look at P1 and P6, if we summate an EPSP and an IPSP, assuming you can see that these are going to be the same strength because these are graded potentials, so they're going to have the strength of the stimulus will change. The strength of will dictate the membrane change. In this case, P1 and P6 have the same strength to their stimulus, and so they cancel each other out. If we look at temporal summation, we're saying, okay, so temporal summation, remember it's two or more signals summated from the same neuron, from one neuron. So we're looking at P1 and P1. There's that time interval there, though, between them where it's not really summated. You have a signal, and then you have another signal. Same thing with the IPSP. You have an inhibitory signal, and then another inhibitory signal. If they're summated, it means that they're fired faster. So you'll have a signal, and then down here you have an inhibitory. So they're kind of riding off of each other. This first one kind of does a little bit of work, and then that second signal gets a leg up from it. Same thing down here. This one has already kind of loosened the jar a little bit. So when the second signal from that same neuron comes again, it can get it can go in even further. Okay, and the all or none law. So we've mentioned this before when we did muscle contractions, and it's a nerve component. It's a nervous principle, not a muscle principle. So if threshold is reached, the action potential generated and propagated down the axon without any loss in intensity. So if threshold is reached, if these signals, either temporal or spatially summated, are strong enough to reach that threshold voltage, then sorry. There's Alfonso. He's sending messages. Where are we? Here we go. If the threshold is not if action potential is generated and propagated down the axon without any loss in intensity. So graded potentials do lose their intensity. They are short-lived. They do not last very long. And depending on the intensity of the stimulus, if you have a bigger stimulus, you will have a bigger graded potential. If you have a smaller stimulus, you'll have a smaller graded potential. Action potentials don't work like that. If an action potential is created, it will be just as strong no matter what. And as it goes down that conductive segment, it will not lose any of its intensity. It will stay the same. If threshold is not reached, the stimulus is considered to be subthreshold, and the voltage-gated channels will stay closed. So there's no action potential. The axon shows the same intensity of response to values greater than threshold. So the book uses the example of a gun. Essentially, what it's saying is that you don't have to, if threshold is negative 55 millivolts, it doesn't matter if you get right at negative 55.0 or if you depolarize all the way to positive 20. As long as you reach threshold, your stimulus is exactly the same. So what they say is, with sufficient pressure on a trigger, the gun will fire. If you do not squeeze the trigger enough, the gun will not fire. But whether you squeeze the trigger just enough to fire, or if you really pull it and you hold it strongly, the firing will still happen the same. The bullet will still leave the gun just as fast, regardless of your strength of squeezing the trigger, as long as you are strong enough to squeeze the trigger. And that's kind of how an action potential gets started. And one of the reasons why you don't have any loss of intensity is because of these voltage-gated channels. Once the action potential is initiated, if the stimulus is strong enough to reach threshold, that's going to start to open up those voltage-gated channels from the graded stimuli. And then because of the change in voltage is enough to open up the channels, it really doesn't matter, how strong or how weak the original stimulus was. As long as it was enough to reach threshold, it will be propagated down. Because once those voltage-gated channels start to open, we'll see in the next segments that it's a domino effect. Once one opens, it kind of causes another one beside it to open, and that will just continue. So the whole important thing is if we reach threshold, we will have this all or none response. If we reach threshold, all of the signal will be propagated down the length of the axon without losing intensity. If we do not reach threshold, there will be no action potential produced.